Welcome students. Today we are going to throw light on sexual incompatibility. As you already know, pollens are received by the stigma and all the pollens are not effective in performing fertilization. The pistil is allowing the pollens of right mating type to function normally and others are rejected. A pistil with functional female gametes is pollinated with viable and fertile pollen capable of bringing about fertilization in another pistil. The two are said to be incompatible and the process is known as sexual incompatibility. There are two types of sexual incompatibility. Number one, interspecific sexual incompatibility, which occurs between individuals of different species. Number two, intraspecific sexual incompatibility, which occurs between individuals of same species. This is also known as self-incompatibility. The incompatibility reactions are under the control of S gene. According to Linskin's 1975, interspecific incompatibility is regulated by more than one gene. It acts as a barrier in free cross pollination and is the basis of creation of new races and species. In incompatibility interspecific crosses, two processes may get involved. Number one, either occurrence of fertilization fails or number two, syngamy occurs, which is followed by abortion of hybrid embryo due to the inadequate development of endosperm. The physiology and biochemistry of incompatibility have been investigated almost exclusively with reference to self-incompatible systems. Now, we will discuss self-incompatibility. Variety of flowering plants are fertilized by the pollens of other plants and not by their own and are known as outbreeders. Self-incompatibility is defined as the inability of the plants having functional gametes, male or female, to produce seeds when self-pollinated. Depending on the origin of factors determining the mating types on the pollen side, two categories of self-incompatibility have been recognized. Number one, gametophytic self-incompatibility, designated as GSI, and sporophytic self-incompatibility, designated as SSI. The difference between GSI and SSI are: first, GSI is determined by the genotypes of male gametophyte itself. For example, Gramineae, Liliaceae, Solanaceae, etc. While as SSI is controlled by the genotype of the sporophytic tissue of the plant from which the pollen is derived. For example, Compositae, Cruciferae, etc. Second point, GSI is two-celled and SSI is three-celled. Third, in GSI, the in vitro germination is easy and in SSI it is difficult. Fourth, in GSI, the respiration rate is low, whereas in SSI it is high. Fifth, the stigma in GSI is wet and in SSI it is dry. Sixth, the rejection reaction in GSI takes place in style, whereas in SSI it takes place on the stigma. Now, physiology and biochemistry of incompatibility. Two reactions have been observed in incompatible process. Number one, a recognition reaction, and number two, rejection reaction. Now, we will discuss them one by one. Recognition reaction. It is the pistil of the flower that has to decide whether a particular pollen grain is to be accepted or rejected. When the pollens comes in contact with stigma, recognition reaction starts immediately in SSI and some GSI systems. Presence of stigma is necessary for the recognition of pollens. For example, in Prunus avium, deposition of pollen grains on artificial stigma are able to germinate and the pollen tube reaches the ovary even in incompatible pollinations. Thus, in such case, the recognition and the rejection reactions are separated by time and space. In GSI systems, the recognition and rejection reactions occurs inside the pistil. Number 2, rejection reaction. The recognition reaction sets in physiological and biochemical processes in the pistil specific to the type of pollen received. In incompatible pollination, the phenotypic manifestations of these changes bring about rejection reaction. The rejection process can occur on the stigma, inhibiting pollen germination or preventing the entry of pollen tube into the stigma, or it may occur in the style inhibiting pollen tube growth, as in GSI systems. Now, genetic and molecular basis of self-incompatibility. East and Mengeles-Dorf, 1925, proposed a hypothesis known as opposition S allele, which deals with the genetic control of self-incompatibility. They were of the opinion that a single gene called S gene with several alleles regulates the incompatibility reactions. Pollen grains that possesses the S allele common to only one of the two alleles present in the cells of the pistil will not be functional on the particular pistil. To understand it further, let us consider a plant possessing alleles S1 and S2 in its sporophytic cells, including the pistil. This type of plant will produce two types of pollens during microsporogenesis. Half of the pollens will contain allele S1 and the other half will contain allele S2. According to the opposition S allele concept, pollens containing S1 or S2 allele never succeeded in performing fertilization in the style of this particular plant, because both the alleles S1 and S2 are also present in the styler region. If the pollens with alleles S1 and S3 were used to pollinate a plant containing alleles S1 and S2 in the styler region, only pollens with S3 allele will bring about fertilization. That means 50% of the pollens containing S3 allele would be functional. The pollen grains with alleles S3 and S4 would be functional on the pistil containing S1 and S2 allele. Here 100% pollens will be functional as none of the allele is common in both the plants. These examples clearly showed that it is the S allele of the pollen which is responsible for incompatibility reaction. In GSI, the self-incompatibility is controlled by the single S allele in the haploid pollen grain. A pollen grain will grow in any pistil that does not contain the same allele. In GSI, all pollen grains will germinate and form pollen tube to grow into the style. Compatible pollen tube will fertilize the egg, whereas incompatible pollen tube stops in the styler region. The growth of the incompatible pollen tube is controlled by the S-Locus, which encodes ribonuclease, sRNAs and S-glycoprotein. The incompatible pollen tube stops growth in the styler region due to the synthesis of female component ribonuclease sRNAs within the style. The sRNase enters the pollen tube and causes degradation of ribosomal RNA inside the pollen tube. This stops the elongation of the pollen tube and ultimately death of the pollen tube results. In S-glycoprotein mechanism, the female determinant is expressed in the stigma. Here the male and female determinants interact with each other and a signal is transmitted into the pollen tube, which results in strong influx of calcium cations. Calcium ions present in the pollen tube are essential for its elongation and gets disturbed by the influx of calcium cations. The pollen tube growth stops within 1 to 2 minutes by influx of calcium cations mechanism. The pollen inhibition by this type of mechanism is reversible. That is, pollen tube elongation can be resumed. In SSI systems, all the pollens of a plant behave similarly irrespective of the S allele they carry. The self-incompatibility phenotype of the pollen is determined by the diploid genotype of the anther in which it was created. This type of self-incompatibility was reported in families Brassicaceae, Asteraceae, Convolvulaceae, Butylaceae, Caryophyllaceae, Stugulaceae, Pulmonaceae. In this system, the pollens will never show germination on the stigma that contains either of the two alleles in the sporophyte parent that produced the pollens. For example, S2 pollens produced from S1, S2 parent will never germinate on an S1, S3 stigma. If a plant carrying alleles S1, S2, the pollens carrying S1 or S2 allele would behave as S1 if S2 is dominant, and if there is no dominance, both will behave as S1 plus S2. In other words, the presence of even one of the allele of the styler tissue in the sporophytic tissue of the male parent would render all the pollens of that plant non-functional with respect to that particular style. S1, S2 pollens would therefore never germinate on the stigma carrying S1, S4, S1, S5 or S2, S3, S2, S4, S2, S9, etc. and will show 100% germination with the plant carrying stigma with S3, S4, S3 and S9. The rejection of the pollens is under the control of S-Locus, which is cluster of 3 loci. Number 1, SCR, S-Locus cysteine-rich protein is a male determinant expressed in anther tapetum as well as in microspore and pollens. Number 2, SRK, S-receptor kinase is a transmembrane protein embedded in the plasma membrane of the stigma cell. SRK has an intracellular kinase domain and a variable extracellular domain. Number 3, SLG, S-Locus glycoproteins is present in stigma and functions as co-receptor for male determinant. When SRK interacts with SCR proteins, and autophosphorylation of the kinase domain of SRK occurs and a signal is transmitted in the stigma. The S1, S2 pollen produces both SCR1 and SCR2 for incorporation in both S1 and S2 pollen grains. If the receptors on the pistil received either SCR molecule, the kinase activates many reactions that makes the stigma functionless for pollen grain germination. Another protein, MLPK of serine threonine kinase nature is attached to plasma membrane and have been reported to have a role in pollen inhibition. Other mechanisms of self-incompatibility. These mechanisms have received less attention by biologists because they are not present in abundance. These are heteromorphic self-incompatibility. Number 2, cryptic self-incompatibility, CSI. Number 3, late-acting self-incompatibility, LSI. Let us discuss them one by one. Number 1, heteromorphic self-incompatibility. This type exists in heterostylous flowers. The features are: first, all the mating types within a species are morphologically distinct and are easily recognized without a breeding test. For example, occurrence of 2 styles, distyle, or 3 tristyle in a flower. The position of stigma and anthers are the main criteria of difference in mating types. The incompatibility reaction is controlled by one gene with 2 alleles, as in dimorphic species Primula, or by 2 genes with 2 alleles, as in tristyle Lythrum. Second, the incompatibility reaction is determined sporophytically. Third, pollen tube growth is inhibited in style region. The only exception is Armeria maritime, where inhibition occurs on stigma. Number 4, dominance between alleles of the incompatibility genes is expressed both in the pollen and the style. In contrast, in homomorphic systems, the incompatibility genotypes cannot be distinguished morphologically, and the incompatibility response relies entirely on physiological mechanisms. Number 2, cryptic self-incompatibility, CSI. In this type of self-incompatibility, both cross and self pollens are present on the stigma simultaneously. Higher seed set have been observed from cross pollen in comparison to self pollen. In CSI, normal fertilization and seed set have been reported through self-pollination even in absence of cross pollens. CSI, now defined as the ratio of crossed to selfed ovules formed when the self and cross pollens are present in equal amounts on the stigma. CSI have been reported in Silene vulgaris. Number 3, late-acting self-incompatibility, LSI. Here, self-pollens show germination and reaches to the ovule, but fails to set fruit. It is also known as ovarian self-incompatibility, OSI. The LSI takes place in 2 ways, prezygotic LSI and postzygotic LSI. In prezygotic LSI, embryo sac deteriorates before the entry of pollen tube, for example in Narcissus triandrus. In postzygotic LSI, the zygote or embryo is not able to develop. For example, species of Asclepias and Spathodia campanulata. Biological significance of self-incompatibility. Selfing of the plants leads to homozygosity and will produce homozygous plants with very low survival value. Self-incompatibility is the way to prevent the homozygosity. Self-incompatibility leads to reproductive isolation and thus made the road for new races and species through the process of mutation and hybridization. When a free flow of genes between the 2 populations of a species stops, they are regarded as 2 variants, and if the difference in the population are fairly large, they are considered to be 2 distinct races or species. Apart from its natural value, sexual incompatibility has some negative impact on plant improvement program. In several plants where anther culture has failed to yield haploids, self-pollination continues to be an important approach to achieve this. In this context, self-incompatibility is a major problem. Methods to overcome incompatibility. The following methods have been proved to overcome incompatibility. Number 1, mixed pollination. In this method, the stigma is disguised from the incompatible pollen by pollinating with a mixture of incompatible and compatible pollens, termed mentor pollens or recognition pollens. To prevent fertilization by the mentor pollen, they are either inactivated by radiations or killed by treating them with chemicals like methanol. Mentor pollen has facilitated to overcome interspecific incompatibility in Cucumis, Nicotiana, Populus, and self-incompatibility in Cosmos, Bipinnata, Theobroma coca, Nicotiana alata, Petunia hybrid. Number 2, bud pollination. Pollination at bud stage have been proved to overcome incompatibility. The incompatibility factor does not reside in the stigmatic exudatus. In the style, the factor complementary to the pollen factor for incompatibility reaction appears only at or just before anthesis. Therefore, if the stigma is self-pollinated at bud stage, when this factor has not appeared, the pollen tube will grow normally and effect fertilization. Number 3, stub pollination. In some plants, incompatibility is restricted to the stigma and certain length of the style. By removing the stigma and the part of the style, the incompatibility have been suppressed. For example, Ipomia trichocarpa contain stigma which showed self-incompatibility and inhibits pollen germination if the stigma and styler part is trimmed and the cut surface of the style is pollinated, germination occurs. Number 4, intra-ovarian pollination. When the incompatibility lies in the stigma or style region, the direct introduction of pollens into the ovary has been proved to overcome the incompatibility. Viable seeds following intra-ovarian pollination have been obtained in several members of the Papaveraceae such as Argemone mexicana, Argemone acroleuca, Papaver somniferum, and Papaver roheus. Number 5, modification of stigmatic surface. Pretreatment of stigma with detergents such as tritone and organic solvents such as hexane modifies or partially solubilizes the pellicle and blocks the incompatibility reaction. Self-compatibility in Brassica campestris could be overcome by application of NaCl to the stigma 10 to 15 minutes before pollination. Number 6, heat treatment of style. Certain plants have incompatibility in the styler region and treatment with temperature up to 50 degrees centigrade overcome the incompatibility. For example, Lilium longiforum treated with 50 degrees centigrade for 6 minutes showed suppression of self-incompatibility. Dear students, this is all about sexual incompatibility. Thank you very much.